Yes, it's Uncle Jegs, and in today's vid...
Uncle, catch the thing.
No, today we're on the field investigation require practical. We need to sample, not catch.
Uncle, you need to use a Quadrat.
Alright, so the Quadrat is out. What do we do now? How should we sample this area?
Dash it over your shoulder. Yeah, it needs to be random, Uncle.
Cool, now what?
Count the thing in it.
If you haven't been able to guess from the intro title of this video, we are looking at the field investigation required practical. Here are the parts. We're going to look at ecosystems, biotic and abiotic factors, field investigation required practical, then we're looking at how to calculate population size and also interpreting kite diagrams. As always, check the description for today's worksheet and the answers to the last week's worksheet as well. Alright, it's time to geek that. The eco part of ecology comes from the Greek word oikos, which means house. Hopefully that helps you remember this long definition. Ecosystem is the interactions of a community of living organisms with their non-living environment. So we're looking at where they live and how they interact with their environment. Let's run through some of these key definitions. A population is the total number of a species in an environment. A community is the group of interdependent organisms in an ecosystem. Let's put all of this in perspective. The population of rabbits here is 6. The population of foxes is 2. The community here is the grass, the rabbit, and the fox. That is because the rabbit depends on the grass for food. The fox depends on the rabbit for food, but the fox also depends on the grass. That is because if something happens to reduce the amount of grass, the rabbits won't have enough to eat, so their population goes down. This then decreases the population of foxes because they don't have enough food to eat. Now, remember our definition for ecosystem. Everything in here is the ecosystem. The rabbit, the foxes, they interact with the non-living water in the stream and also the shelter in the cave. Also, the plants interact with the non-living sunlight via photosynthesis. I'll save food chains, food webs, and bioaccumulation for a later video. There are 2 types of things that can increase or decrease the population of an organism. They are categorized as biotic or abiotic factors. Biotic factors, like biology, is all about living things in the environment. There are 4 biotic factors that you need to be aware of. The first one is the availability of food. The higher the availability, the higher the population. The second is new predators arriving. The higher the number of predators, the lower the population. The third is the number of new pathogens arriving. Pathogens are microbes that cause diseases, so the higher the number of pathogens, the lower the population. And finally, the competition by other organisms or species. The higher the competition, the lower the population. I'll break down the different types of competition in a later video. There are 7 abiotic factors that you need to be aware of. These mainly affect plants or aquatic animals. The first is light intensity. The more light, the more photosynthesis. The second is temperature. Remember our required practical on enzymes and optimum temperature for that one. Click the link to go watch that. The third is moisture levels. This is absorbed in the roots and released in the leaves via transpiration. The fourth is pH and mineral content. Remember, this also affects the enzyme activity. The fifth is wind intensity and direction. Strong winds can damage your plants. The sixth is carbon dioxide levels, and that's for plants. And the seventh, the last one, is oxygen levels, and this is mainly for aquatic animals. Here are some summary notes on all of that. There are two different practicals for this section. Each requires a different technique. You could either investigate the population size of a plant species using random sampling, or investigate the effect of a factor on plant distribution using a line transect. Both types of sampling methods have their advantages and disadvantages. An advantage of a random sampling is that it's pretty easy to implement. A disadvantage is that it can be imprecise if your population is heterogeneous. Heterogeneous means that the population is not evenly distributed across your area. Imagine you had bare plantain over here, but hardly any over here. If you did your sampling, you might get a very inaccurate estimation. A line transect is an example of systematic sampling. The advantage of systematic is that you have a maximum distribution or dispersion of samples throughout your population. However, a disadvantage is that it is more susceptible to personal biases. Here are the steps for random sampling. The first thing, you need to use a random technique to pick where you're gonna sample. This could be you picking the coordinates from a bag that your teacher has already prepared. It could be you using a random number generator, or if all fails, you dash it safely over your shoulder. The second thing that you have to do then is that when your quadrat is on the floor, you count the number of your chosen plant species inside the quadrat. Then the third thing you do, you repeat these steps 10 times. Now to calculate your population size. We use this equation to estimate your population size. Your estimated population size is your total area divided by your area sampled times your number of organisms. Remember that the area of a square is the length times the height, and the size of my quadrat is 1 meter, so that gives me an area of 1 times 1, which is 1 meters squared. Since I'm using my quadrat 10 times, my area sampled is 10 times 1 meter squared, which is 10 meters squared. Now look at my field. 10 quadrats can fit the length and also the height. So the total area of my field is 10 times 10 meters, which gives me 100 meters squared. 100 meters squared divided by 10 gives 10, and that times the number of plantain gives the total estimation of 220 plantains. The next kind is a line transect. The most likely factor that you would measure is light intensity, but the exams could give you any of the abiotic factors that we looked at. First thing you do is you first measure out 30 meters of a measuring tape from the base of a tree to an open area on the field. The second thing you do is that you place your quadrat along the transect at these specific distances. So I'm going up in 5s. Then you count your number of plants using your quadrat, and after that you use a light meter to measure the light intensity at each area. Then you record your results in a table like this one. Now this next bit is a higher level skill. We're going to use this table to draw a kite diagram. This will also help you understand how to interpret kite diagrams if you ever get some in your exams. The first thing I do is look at how long my distance is. In my example, I've used 30 meters, so my scale on my x-axis has to go up to at least 30 meters. The abiotic factor goes directly below the number of organisms that you sampled. The highest light intensity is 10, so I make sure I have enough space to plot this. As you see, the further the distance, the larger the light intensity. That's because we started off near trees that obscured the light. Now for the tricky part. A kite diagram is a symmetrical diagram showing how much of something you have in a specific area. So you first start off with a guideline, which is at your 0 point, and you need to have, let's say for example, I had 10 plants at a specific area. That would mean that I need to have 5 points above the guideline and 5 points below the guideline. So let's apply the thing then. I start off by checking the highest number of my plant, that's 30. So I then need to make sure that I have 15 above my guideline and 15 below my guideline. If I go up in 5s, that means I'll need 7 marks because I also need a mark for the 0 point. Now look how it's done. At the 0 distance, the number of plants was 0. It's the same with when my distance was 5 meters. But now look at when it's 10 meters, I have a number of plants as 6. So I have 3 above and 3 below my guideline. At 15, it's 5 above and 5 below because the total was 10. I carry this on until the end. When I finish doing that, I join up all my points on either side of my guideline. They may give you a kite diagram to interpret. In such situations, you will likely be given a key to interpret the number of squares and what that means for your number of organisms. So in my example, 4 squares is equal to 10 individuals. The first thing to notice is that as the light intensity goes up, so does the number of plants. A question you may get is something like this: how many individual plants were they in 20 meters? So you count the squares. I have 8 squares at 20 meters. If 4 squares equals 10 individuals, 8 squares then must be 20 individuals. There we go. The field investigations require practical. That was a lot of information, so go back and rewatch if you need to. Check the description for this week's worksheet and the answers to the previous video's worksheet. Like if you like, subscribe if this is necessary. In a bit.